This is a podcast on the differences between DNA and RNA, of which we should know that there are three. The first is that DNA will form a double helix. It's a double-stranded molecule. So there are two DNA strands in DNA, and there's only one in RNA. The second is that in the ribose sugar, which is present in the nucleotide, remember that's our nucleotide monomer, the difference between the carbon 2 and carbon 3 groups on them is different. So we can see here, in ribose, we have two OHs, 1 and 2. So there's 2 times oxygen. This is the ribose sugar. Deoxyribose means that, and the key term here is deoxy. The deoxy, if you use deodorant, you take away the odour. If you decontaminate something, you take away the contamination. If you delouse, you take away the lice on your head. So D here means to remove. And so we've removed an oxy from the ribose. And so here we can see that instead of OH on carbon 2, we actually have just H. So there's a missing O. So we have deoxyed it. So ribose has 2 oxygens and deoxyribose only has 1 because one of the oxygens has been removed. Number 3 is the bases that are used in base pairing, and there's just one difference, and we don't need to worry too much about the chemical formulas here, but the key is that we have thymine for DNA and uracil for RNA. So that is thymine for DNA and uracil for RNA. They will base pair obviously with adenine, which stays the same. So, number 1, double-stranded versus single-stranded. Number 2, only 1 oxygen in DNA because we've been deoxyed, as opposed to 2 oxygens in the 2 H groups on RNA. And number 3, we use uracil in RNA instead of thymine. Unos, dos, tres.